And we will move on to the next short talk by Johanna Perens from the Technical University of Denmark. And she will tell us about multimodal mouse brain atlas with a stereotaxic coordinate system for bridging in vivo and ex vivo 3D imaging techniques. Please, Johanna.
Thank you. And also, thank you for the possibility to present my work and the work also of the company I'm actually at. It's a big pleasure. Yes, so I'm a research scientist, actually now after finishing my PhD at the Danish biotech company called Gubra. And this work was done during my PhD time. I was an industrial PhD student at Gubra, and it is done then in collaboration with the Technical University of Denmark, but also the Danish Research Center for Magnetic Resonance. And I will present you then today a multimodal atlas for combining different 3D imaging modalities. So Gubra is a contract research organization where we perform different preclinical studies for pharmaceutical companies, but also academic university groups. And Gubra is focusing mostly on metabolic diseases, including obesity and diabetes, and also comorbidities of these diseases. At Gubra, we have a 3D imaging team. We have a light sheet fluorescence microscope, actually 3 of them even, and we perform high throughput whole organ imaging. And we do all sorts of organs, from brains to kidneys to livers, everything. But the big interest is the brain, of course. And here's the one data set, how our brains look like. And very often we do brain activity measurements. So here you can see a CFOS protein, which is expressed in response to a drug called semaglutide. It's a body weight lowering compound. And we can image not only brain activity, but also drug distributions or neuronal populations, receptors, and also, of course, axonal projections, etc. Just a small reminder. So we do light sheet fluorescence microscopy in ex vivo, not in vivo. And the workflow we have is that we extract perfused brains from the skulls. We do whole mount immunolabeling. We clear the brains. And then we use light sheet microscope to optically section the brain and collect our 3D data. In case, if you have endogenous fluorescence, you can, of course, just skip the immunolabeling step and do immediately the clearing. So there are a few limitations which arise with clearing the tissue, or actually with the whole protocol. And it depends of different chemicals you are using. And these limitations, or there are limitations for the image analysis actually, not imaging itself. But for example, if you do analysis and you actually want to see where your signal is and count or quantify that signal, then you need to know in which brain region you are. Okay, there are existing atlases, but region delineation is a little bit difficult in these brains, and it is because these chemicals used for clearing, some of them, are inducing deformations in the brain. So it means that it's hard to use existing atlases to delineate the regions. It is also difficult to identify the signal localization or signal in your light sheet fluorescence brains, or in the signal you measure in these brains. What is the real localization of these in a living brain? And it also means that if the brains are deformed, it's hard to map them to the other 3D imaging modalities, such as MRI. And we developed the multimodal mouse brain atlas for integrating different imaging modalities. And here is a workflow. How did we create the atlas? It's a little bit different atlas because the concept we use is different from what is used usually. So we don't put all the brain templates, so atlases also always contain brain templates. We don't put them in the same space, in the same morphological space, but they're in their own morphological spaces. And what we did is that we created we created 2 brain templates, one for MRI, one for light sheet, and we bridged them using existing Allen mouse brain atlas, which is a very well-known and often used atlas, which has brain region annotations or delineations. And then we also did CT imaging of mouse skulls to extract bregma and lambda positions, because these positions are used to create the coordinate system, a stereotaxic coordinate system. And at the end, when we bridged these different brain templates using deformation fields, so you can move between the spaces and transform your data between the spaces using these deformation fields. But we could also use these deformation fields to bring the region delineations from Allen to MRI and light sheet, and the coordinate system then from MRI, because it was made in the MRI space, to the Allen and to the light sheet. First of all, I will tell you a little bit about how we did the light sheet template and the atlas. And why do we use this interesting concept of having them in a different morphological spaces, and why don't we use just one space? And the reason for that is, so if we acquire new brains, here one brain, we would like to map it to the atlas. So if I now do a whole brain registration, so rigid, affine, and nonlinear registration to the Allen mouse brain atlas here, then it's fast, because it's a whole brain registration, but it's inaccurate. We have inaccuracies in the hindbrain regions, especially areas like area postrema. What we want to have is a fast and accurate registration, because we do high throughput imaging. So for that case, we would like to have a light sheet template, which is like an average light sheet brain. And we would like to have a fast registration, but we want to get accurate region delineations. So how we do it is that once we do a registration between Allen and light sheet template, and it's a multi-regional registration, so we register bigger brain regions to the light sheet template and bring then the small brain regions within them together with these deformations. I can show you later on. Yes, and we do it once. So here is a workflow for generating a light sheet brain template. At first, we collected 139 images of, there are high-quality images of whole mouse brains. We made a brain template by iteratively registering and averaging them. And here you can see one of the coronal slices of this template. And then we did a multi-regional registration between the Allen and the light sheet template, and then brought, so to say, all region delineations from Allen to our light sheet. And here you can see the result for the coronal section. We also validated that the mapping of several brains, here we used 10 brains, to map these brains to the light sheet template is a lot more accurate than by mapping them to the Allen template. And we did it by evaluating a metric called intensity variance. And we calculated intensity variance for every brain region. And we found out, so the higher is the intensity variance per brain region, the less good is your registration. So you can see that specific areas, like hindbrain areas, but also in some interbrain areas, it is actually, the registration is worse because the intensity variance shows that it's worse for Allen and better for light sheet. Yes. Next, we took 12 mouse brains, mouse heads actually. We imaged them with the CT. We imaged them also with the MRI, and made a brain template in MRI space with the coordinate system. So, because we did the CT and MRI from the same brains, we could co-register the CT on top of the MRI brain. and then extract the skull landmarks from every single skull, which is in MRI space. We extracted the skull surface, projected it on a one plane, annotated the sutures, so there are 3 sutures, and then fitted them and found the intersection points, because intersection points of these sutures here are called bregma and lambda, and these are used generally for generating atlases. The Z position of these bregma and lambda positions were just defined as being on top of the skull. So now we have bregma and lambda points for every individual brain. And the next step was to generate the MRI template. MRI template was generated the same way as the light sheet template, iteratively registering and averaging. And then we also mapped every single bregma and lambda point, so the 12 points, in total 24, mapped them to the light sheet template. And then we averaged them in the MRI template. And then we averaged them in the MRI template. So here is the result for the MRI template. You see the template here, here on top you have a bregma, and here you have a lambda point. And why do we have 2 points? So one of them is used for being the origin of your coordinate system, but you need the lambda point, the second point here, to actually find the correct angle of the brain also. Yes, and then we could also measure actually the variation of these average points, bregma and lambda, and this variation varied from 50 microns to 150 microns. It's quite a lot, and it actually shows how important it is to use many brains to collect the bregma and lambda points and average them, and based on these average points, make a coordinate system. So now, in that point, we have MRI template with a coordinate system, because you can generate, based on bregma and lambda, a coordinate system. We have an Allen mouse brain atlas, and we have a light sheet template with Allen brain regions. So now we generated deformation fields between these templates, so in total 4 deformation fields, and used them to map the coordinate system from MRI to Allen and to light sheet, and region delineations to the MRI and to the light sheet. And here you can see the coordinate system in all different spaces and all different coordinates, so x, z, and y here. The colors here indicate the coordinate values. So the pink is for positive and the green for negative values. And the system was generated according to the convention of Franklin and Paxino's atlas, which is one of the few atlases which actually has coordinate system. And it's a histological atlas. And the stripes you see on the image, it's like a contour plot. So basically every stripe you see indicates the same coordinate value. So because the coordinate system was generated in the MRI space, of course the stripes are linear. But if you now look at the Allen atlas, you see that these stripes, the coordinates start to bend the contour values. And also in the light sheet space, these coordinates are bent even more, showing that there are deformations actually happening. But now we have in vivo coordinates, because MRI is an in vivo space. We have put them in a light sheet space. We also validated the accuracy of that atlas by checking if every single coordinate, if these coordinates actually match the same anatomical structures. And we also tested the deformation fields by taking just one random MRI brain and using it with these deformation fields. We put it to all different template spaces. Sorry. So just want to show that. So here, this MRI brain we mapped is shown in gray, and the different templates have different colors. And you can appreciate that actually all the brain structures are matching. So we can put MRI brain to all the different spaces until the light sheet space also. Yes, and this is our final atlas. We have 3 templates. We have in every template the regions. We have coordinate system in all the template spaces, and the deformation fields to map between these spaces and bring our data. In conclusion, we have the multimodal atlas, which permits optimal and fast mapping of new data samples, and also integration of in vivo MRI, but also ex vivo light sheet data. We have modality-specific templates which are bridged using the Allen Atlas, and it has very extensive resources which can still be integrated in the atlas and used in that atlas space. We have a coordinate system based on average skull landmarks. It allows accurate signal localization and also stereotactic inventions. And finally, this atlas is extendable with all different kinds of templates and coordinate systems and delineations, whatever you would like. We can add it to that. Yes. And finally, I would like to thank the scientific team at Gubra, the professors Anders Bjørholm Dahl and Carsten and Tim Derby, and finally also BrainGlobe. So BrainGlobe has made this light sheet atlas available for you already, and the multimodal atlas will also be available for use for everyone there. Thank you.